Welcome to tonight's important lecture. Alessandra and I were just reflecting on stem cell biology. Just 10 years ago, this was an idea that many pioneers were exploring in labs around the world. And incredibly today, there is stem cell therapy in clinics around the world, treating major unmet medical conditions. Patients, we all have family and friends who suffer from maladies, age-related degeneration, someone with a heart attack or heart failure, COPD, massive inflammation, single pathway biologics, the kind of standard of care, the way we did things forever and a day. And now we have something incredible in our hands, the power of the living cell. It's innate in all of us. Google is an amazing tool. I was asked to introduce to you tonight's guest, and of course, humbled as a California boy myself, thrilled to know that there's an extraordinary person amongst us who's a pioneer, who took hold of the concept, the power of the living cell, and made it a mission to help bring it forward to its place where it is today and where it will become a standard of care in the years ahead. I won't do justice to her extraordinary career, but I will say, from a place of humility, as a patient and someone who's very passionate about seeing the power of the living cell become ubiquitous as a standard of care therapeutic across many disease states. It's an honor to introduce Dr. Helen Blau to you tonight. We have an extraordinary evening ahead of us with her lecture. And with that, I will just simply turn the floor to a colleague who can give you an extraordinary experience about this extraordinary woman who you're going to hear from tonight, Alessandra.
It's a pleasure for me to be here and to be introducing our speaker for this forum, Helen Blau. Helen is a professor at Stanford University and is the director of the Stem Cell Regenerative Center. And during her career, she received many prestigious recognition, and I'm just going to mention one. She's a member of the National Academy of Science since a few years. I had the privilege of training with her, so it has been an amazing experience for me as a postdoctoral trainee, and I consistently saw her capacity of actually consistently bringing innovation to stem cell biology field. So during her career, many years ago, she pioneered studies in trying to understand the plasticity of a cell, and really like, how can we define a cell identity, how is it maintained, and how can we manipulate it? And she did very pioneering studies through cell fusion to demonstrate this fundamental process. And then over the years, she really made major contribution to the skeletal muscle stem cell field, and not only, by really defining and trying to understand how muscle stem cells are actually regulated and how can we actually utilize them as tools to treat muscle degenerative diseases, and also how can we actually improve or prevent tissue aging from happening. And she has been, for me, an amazing mentor and a constant inspiring example of really how keep asking critical questions and trying to find innovative and creative answers. And she has been a major member and part of the stem cell biology field. And I'm really happy that she's here tonight. And I would like to introduce her. Thank you.
Okay, well, it's my great pleasure to be here. And the first thing I'd like to do is give a shout out to CIRM and Prop 71 for having made a lot of the work I'm going to tell you about possible, and much more than that. It's really been a fantastic source of funding, inspiration, and support. What I'm going to tell you about today is rejuvenation of stem cell function in order to combat muscle weakness with aging. No pain, no gain. First, I have a disclosure. I have a financial interest in Myoforte Therapeutics, and part of this presentation has been patented and licensed by that company. So, aging. Aging, as depicted by Edvard Munch, is typically this relentless progression. One envisions the young woman who is vibrant and then matures to a full-grown woman, and then middle age, and then decrepit aging, as if it's this relentless progression and quite depressing. But does it have to be that way? Well, if you look at the statistics, we're living longer, but are we living better? Health span is not keeping a pace with lifespan. And one of the goals of my lab is to improve that health span, the quality of life. And the way we're approaching that is through rejuvenating muscles and improving muscle function so that you can do all the things you want to do. So you can dance and run and ski like this woman here. And muscle is central, of course, to everything you do in life. Whether you're a dancer with the ballet or Rodin's thinker, you're using your diaphragm muscles to breathe. And if you're Steph Curry of the Golden State Warriors, you're using every muscle in your body to shoot those three-pointers. Muscle is very important. It's the largest tissue in your body. It's 40% of the muscle mass, body mass, and it's the most regenerative of the muscle tissues, and its function declines with aging. Muscle repair is crucial to your muscle function and quality of life, and that repair or regeneration is fueled by stem cells, the muscle stem cells. And these stem cells reside along the muscle fibers, and they're in a quiescent state until there's injury, and then they're induced to be activated, self-renew, expand, commit, and differentiate. So what happens with aging is that your muscles change, and your regenerative capacity progressively declines. And you see that the muscle tissue ends up full of fibrosis and fat, and you're less strong. Well, we wanted to understand the nature of these stem cells. People had defined them anatomically, but didn't know of them functionally. And this key work was done by Alessandro Sacco, who just introduced me. And I have to tell you, there is no greater pleasure in life than to be introduced by your scientific child who has successfully gone out into the world and become an associate professor and renowned in her own right and publishing extremely well. She's a fabulous scientist, and I am just delighted that she introduced me today and that she's here. So Alessandra did these experiments, and she proved that we had isolated the quintessential, the stem cell, using criteria that had been established by hematopoiesis. And that was done using this workhorse, the GFP luciferase donor mouse. It's a transgenic. Every cell in its body expresses green fluorescent protein and luciferase. And I'll tell you why that's so important. So you can isolate these stem cells, and then we would inject them into the injured muscle of a mouse, and then look at how well they engrafted and transplanted. And we did this as a single cell analysis, and that was only possible because we could measure the output, the proliferation, the progeny generated by a single stem cell by using a CCD camera to measure luciferase. And that's what's shown here. So she injected 72 mice with a single cell, and you can see that 3 out of the 72 engrafted, gave rise to lots of progeny that expanded, and they were able to commit and differentiate into muscle fibers, shown here in green. And she did that twice, 3 out of 72 again. And that proved that we had the stem cell. It could self-renew and differentiate. And then we used this approach in almost all subsequent experiments because it is so potent as a way to monitor stem cell function. And one of the first challenges we were struck by there, we had the stem cell, but as soon as we put it in culture on typical culture plastic, the stem cells no longer behaved as stem cells. They lost their stem cell function. So when we took fresh muscle stem cells, we saw an exponential increase when we injected 5,000 of them, and then it plateaued. That's an interesting thing about muscle, actually. It knows when to stop. The cells are not tumorigenic. There's something about that environment that is non-tumorigenic and safe. The cells know when to reach homeostasis and stop proliferating. By contrast, when we took cells, many more cells that were cultured on plastic and we injected them, we saw a decline, an actual decline, an order of magnitude decline in stem cell engraftment. So we couldn't study them. So that was a challenge. And a group in my lab, multidisciplinary group, which is the way I like to bring people together from different disciplines. And so Penny Gilbert, a biologist, Karen Havenstreit, a chemical engineer, Klaus Magnussen, artificial intelligence expert, and Ben Cosgrove, a bioengineer, got together and thought about how the stem cell might see the world. And we call this niche in a dish. And looking at the microenvironment. And maybe the cells didn't see anything quite that stiff. So what they did was to measure the rigidity of muscle tissue, did rheometry, and determined that the tissue is 12 kilopascals. And when you look at that by comparison with tissue culture plastic, you can see that the plastic is 5 orders of magnitude more stiff than muscle. So the cells never, ever in the body experience anything that stiff. So we wondered whether maybe we could maintain them if we recapitulated the rigidity, the elasticity of the body, of the muscle. So we fabricated hydrogels that spanned the rigidity going from that of brain, 3 kilopascals, all the way through muscle, bone, and to the rigidity of plastic. And when we isolated the cells from our GFP luciferase mice using the cell sorter, and then cultured them on these different rigidities of substrate, 12 being muscle, and then we allowed them to grow for a week and then transplanted 100 cells and measured their engraftment by bioluminescence imaging. When we did that, what we saw was it made a huge difference. You can see here these are individual legs and how well the cells engrafted. Here's an example, and you can see that on 12 kilopascals, which is the rigidity or elasticity of muscle, the cells engrafted robustly. And here's for large numbers of mice, and 50% engrafted if they were grown on something that was the rigidity of the muscle tissue. And that rapidly fell off if it was softer or more stiff. So stem cells care. We wondered then whether there was a difference between young and aged. And we compared them by injecting just 10 cells. And we found that if we took the young, they robustly engrafted, and the aged lost that capacity. And that was shown by bioluminescence and by histology. When we did a limiting dilution, we discovered that, in fact, 2/3 of the cells were non-functional in the aged. So we sought to find, now we had a hydrogel platform where we could maintain the cells in culture. We sought to find drugs that could rejuvenate their function. And we did a candidate drug screen, and we found that SB202, this is a drug that targets P38 MAP kinase. It led to vast proliferation in culture. And we reasoned that maybe that was rejuvenating the stem cells. You can't tell from just looking at numbers, so we had to do an in vivo study. To see whether this was a reasonable target, we analyzed the aged cells and found that they had elevated phospho-P38, which is the target of the inhibitor. And the young do not, so it seemed reasonable. So we then cultured them in vitro. We took the cells from our GFP luciferase mice from young and from aged animals, and we isolated them and put them in culture either on plastic with the drug or on hydrogel with the drug, and then ascertained whether they engrafted. And what you see here is that the young are doing so well on the hydrogel, the drug makes no difference, and the aged are doing so poorly on the plastic that the drug doesn't help. But when you have the aged cells on a hydrogel treated with the p38 MAP kinase inhibitor, you get robust engraftment and a dramatic increase. And so you can see it made not much difference with the young, but with the aged, we saw robust increase in engraftment, so that it was on par, in fact, with the young now in terms of engraftment. But we wondered what that meant in terms of function. So, of course, the ultimate test for function is to see how well the cells lead to an increase in strength. So we took cells from an aged donor, and we cultured them on the hydrogel with the SB drug, the p38 MAP kinase inhibitor, and then transplanted them into the injured muscles of another old mouse. We did this for a number of mice. And what we found is exemplified here. So the twitch force, which is a measure of muscle strength, was quite high, and it's about 50% of that for aged mice. And if they're injured after a no-texin injury, then you see that strength really falls off. But if we transplanted the progeny of 200 cells that had grown on the hydrogel together with the SB drug, we got robust engraftment on par with young. And this is shown for a number of mice now, that we were able to restore strength of the aged mice to that of a young mouse. So that was very exciting. However, this drug, SB, turns out to have many off-target effects, and so we wanted to find something else. It showed us that this was possible, rejuvenation of function. So what we found was that the aged muscle cell population is intrinsically different from young. It has reduced regenerative function, high p38 signaling, and elevated levels of p16 and p21, which are senescence markers. And we also found that biophysical and biochemical signals synergize in order to rejuvenate and expand the aged muscle stem cell population. But as I said, this drug, the p38 MAP kinase inhibitor, SB, has been in the clinic, and it's been found to have off-target effects, and so it's not actually useful. It's useful in principle to tell us that this is possible. So we decided to seek extrinsic regulators of muscle stem cell function and see whether we couldn't find something that might be more clinically useful. And we turned our attention to these facts. So we knew that one of the first things that happens after an injury is a wave of inflammation. And this is followed by an increase in fibroadipocytes and fibroblasts, and then finally a transient increase in muscle stem cells. The important thing to note is that these waves are transient. They're not chronic, they're not sustained, and that appeared to us to be critically important. So we turned our attention, and we are Andrew Ho and Adelaida Paya in the lab, who are very talented scientists. And we turned our attention to this inflammatory wave, and we reasoned that maybe the stem cells would be exquisitely sensitive to something in that inflammation. So to address that possibility, we did a bioinformatics screen, an in silico screen. We analyzed the transcriptome of cells that had been activated following injury. And we compared our transcriptome with that of two other labs, Buckingham and Fukada. And we got the usual suspects, the cytokines, and then some inflammatory markers. And we were struck in particular by this one, known as EP4. What is EP4? EP4 is the receptor for prostaglandin E2. So we reasoned that people had not noticed EP4 before because they had not noticed prostaglandin E2 because you would not detect it in a transcriptome analysis. It's a metabolite, and it's generated by phospholipases from the membrane to yield arachidonic acid, which is then metabolized to several intermediates, giving rise to prostaglandin E2. And this metabolite, this inflammatory metabolite, is very potent and acts on 4 different receptors via different second messengers. And what we found is that on the muscle stem cells, EP4, this one receptor, is specifically expressed. Notably, non-steroidal anti-inflammatory agents, or like indomethacin, are inhibitory of the synthesis of prostaglandin E2. And I'll come back to why that is important. So prostaglandin E2 is an inflammatory mediator, and we wanted to know whether it was, we saw it on this transcriptome analysis, was it actually part of that inflammatory wave? So we did a notexin injury, and we analyzed PGE2 in the muscle tissue lysates, and we found that, in fact, there was this transient increase in PGE2 that completely paralleled that wave of inflammation. And the prostaglandin E2 synthesizing enzymes also followed that wave of inflammation. And if you looked histologically, we also saw that it accumulated at day 3 and was gone by day 6, fitting with this transient rise. And a source of prostaglandin E2 is its niche, the muscle fiber. These muscle stem cells lie along the muscle fiber, and the muscle fibers, when we isolated them and put them in medium, we were detecting PGE2 in the medium, in the conditioned medium. So one of the sources of PGE2 is the muscle fiber which they lie against. And we proved that by treating these muscle fibers with indomethacin, which inhibits prostaglandin E2 synthesis. It's not the only source. It turns out that macrophages and endothelial cells also produce PGE2 and are part of that wave of inflammation. So PGE2 works on the EP4 receptor, and we found that the stem cells only express the EP4 receptor, none of these other receptors to which PGE2 can bind. And we found that it's induced in response to PGE2, so the levels become higher on the muscle stem cells after they're exposed to the inflammatory metabolite. Notably, it's expressed highly on fresh muscle stem cells after injury. Once they're in culture, they lose it, and on myofibers, it's not expressed at all. So it's quite specific to the stem cell in the muscle tissue. So how does it work? PGE2 promotes the expansion of the stem cells in culture. It induces them to proliferate. And we thought that since it's present acutely, it's just transiently in vivo, that maybe it would function in that way also in vitro. So we exposed the cells, the isolated muscle stem cells, to PGE2 for 24 hours, and then examined cell numbers a week later. And we saw that this brief exposure to the drug was sufficient in order to induce a robust increase in the cell numbers. And when we did time-lapse microscopy, we found that the time to first division was greatly accelerated and proliferation increased, as shown here for the generations that are generated by time-lapse microscopy. So PGE2 is inducing robust proliferation, cell division, rapid division of these stem cells. And we found we wanted to see what its target might be that might be inducing this cell division. And we did a transcriptome analysis after PGE2 treatment, and we found NER1 was the top candidate or target. And when we looked in tissues after injury, NER1 followed exactly the time course of PGE2 and of the injury response of inflammation. And if we isolated the muscle stem cells and we treated them with PGE2, again, we saw that NER1 was strongly induced. So NER1 appears to be the target, and NER1 is known to act on a number of cell cycle regulatory genes, and so it's causing the cells to proliferate. And in a fascinating way, the cells have a memory for this molecule. They see it very briefly for a 24-hour period, and in fact, what we know now about this molecule is that it's around for about a minute. It has a very short half-life, And the cells have a memory. So we're now in the lab resolving that. What is this epigenetic memory? How has this changed? So that the progeny, for weeks afterwards, remember that they saw this drug, because it robustly changes them. So we wondered, would this work as a cell therapeutic? Would it enhance the function of the cells in a transplantation study? So we cultured the cells. We isolated them from our GFP luciferase mouse, and we cultured them in the presence of PGE2 for 24 hours, and then we transplanted them into injured limbs of mice. And what we saw was this robust increase in engraftment by about an order of magnitude over the controls. So the cells were able to keep proliferating and remembering that 24-hour exposure in vitro, and continued to proliferate and engraft. But as before, they know when to plateau, and they don't continue. And then we wondered, could we bypass the culture step? Maybe because the activity of this drug is so short, maybe we could just co-inject the drug along with the cells. And so we tested that possibility. We co-injected isolated muscle stem cells together with PGE2, so no culture at all. And what you can see here is that they engrafted even better. They robustly regenerated the muscle tissue. And to see whether they were still stem cells, we did a second injury with no texin. And because you see this increase, that indicates they are still stem cells. Because if they were progenitors or more specialized and committed, they wouldn't be able to respond again. But they were able to kick in a whole nother round of active proliferation for many, many progeny. So this was a really robust response. And I've studied a number of regulators over the years in my lab, and I've never seen anything quite as potent as that. So this stands to be a very strong way to do a cell-mediated therapy. But we wondered, maybe we could bypass that step. Maybe we could even stimulate the stem cells that are present in the muscle tissue and get them to perform better without doing a cell therapy. That would be less costly, less cumbersome. It would evade the whole GMP facility need by simply delivering this molecule to muscle tissues. To test that, we had to make a whole new mouse model. And here what we did was to make a transgenic that is a PAX7 mouse. PAX7 is the hallmark transcription factor of the muscle stem cells. And we had it drive luciferase, so we could read out the stem cell numbers in terms of light as before in a CCD camera. And we did that by tamoxifen in order to flux it and activate this transgene. So we could read out PAX7 luciferase. We injected the cells, and then we into an injured mouse, and we just injected the drug, this time no cells, just to see whether the drug alone could stimulate the stem cells. And what you see here by bioluminescence is a twofold increase, which is very robust. And that's shown here by bioluminescence imaging. So it is a potent inducer of the stem cells that are residing in your muscle tissues. So we wondered how important this signaling pathway is. How important is prostaglandin to the stem cells, to your muscles? No one had really studied it before. There were some old papers where people added it to progenitors and saw they divided a little bit, but there was nothing that characterized its role in muscle. So what we did was we generated another mouse where we could flux out or genetically eliminate the receptor, EP4, on muscle stem cells specifically, and look at the effects. And when we did that by Cre-mediated fluxing, here are the controls. You have cells, and they have the EP4 receptor, and you add PGE2, and you get a robust increase in cell numbers. However, the cells that are lacking the receptor, you can add all the goodies you like. You add PGE2, there's serum there. They're hardly growing at all. So this seems to be absolutely crucial for the viability, for the division of these cells. You see cell cycle arrest. The cells are really not dividing. And if we transplanted those cells, here we see that the controls are nicely engrafting, robustly giving a signal, but the ones lacking the receptor are actually declining. So they're unable to engraft, they're unable to repair the damaged muscles. So this pathway just appears to be extremely crucial to the stem cell function. Well, to look at it further, we wanted to know not just about engraftment by imaging, but again, the most important measure, which is strength. And so here we eliminated the receptor in young adults, and then we performed an injury, and 4 weeks later, we ascertained the strength. And lo and behold, what we found was those mice that lacked the receptor lost strength. So if they couldn't regenerate because they lacked this receptor, which is so crucial to the function of these stem cells, they became weaker, both in twitch force and tetanic force. And that's shown here for a large number of mice becoming weaker. So really, it's the only pathway I know of that is so crucial to the function of the stem cells and for repair of muscle. Without it, you actually get weaker. And to look at it in yet another way, what we did was to take our Pax7 luciferase mice and then treat them after an injury with non-steroidal anti-inflammatory agents. That's like ibuprofen. And what we found is you got a decrease in engraftment. So you're blocking the synthesis of PGE2 by the tissue, by the injured tissue. And now you see that you're getting a decrease in stem cell function, and lo and behold, you see a remarkable decrease in strength as well. So if you're missing the receptor, or if you block the synthesis of this metabolite, mice get weaker. So that was about a year ago, and it was picked up by the New York Times, and they wrote, "Bring on the exercise and hold the painkillers." And I'd say that's something you should think about. Next time you work out or you go for a jog and you feel achy and you're reaching for that ibuprofen, maybe you should think twice, because this really shows that you can actually undo all the good you were doing with your exercise. And it's known that these NSAIDs bind irreversibly to COX-2 and COX-1, so that the effect is extremely long-term. You rely on de novo synthesis of those enzymes to make more PGE2. So what I've told you about till now is that we've discovered a new mediator of stem cell expansion that is playing a major role in strength, prostaglandin E2. And after injury, the PGE2 signaling, so this is an endogenous response. This is a natural healing mechanism that we're capitalizing on, because when there's damage to the myofibers, PGE2 signaling is induced, and that leads to stimulation of the EP4 receptor and cyclic AMP generation and activation of NER1, which then turns on all these downstream factors that are involved in proliferation and expansion of the stem cell population. And in contrast, if you block that pathway, either by lacking the receptor on the stem cells or by blocking the synthesis of prostaglandin E2 with NSAIDs, then instead of robustly increasing muscle function, you in fact get a decrease in repair, and you impair regeneration, and you have reduced strength. So what I've told you about till now is all in the young. And interestingly, we weren't able to make the young any stronger because they regenerate so well. What we were able to show is just how crucial this pathway is in the young, and that when you lack it, you get weaker if you have an injury. But we wanted to know what's happening in aging. What will this help with the decline in function that's associated with aging? So as I told you, in the young, you get these nice waves of response once there's an injury, and inflammatory cells come in, and then fibroadipocytes and fibroblasts, and eventually the stem cells are activated. That's in the young, healthy niche. In aging, it's different. You have this chronic inflammation, chronic high levels of fibroadipocytes and fibroblasts, and it's really not this nice transient wave. So it was not obvious that PGE2 would work in this context. And when we looked at the tissue after an injury, and we measured PGE2 levels in the damaged tissue, what did we see? We didn't see this nice little transient spike as we did in the young. We saw chronic high levels of PGE2. And when we measure strength in the aged, what you see is that aging, your strength declines on this twitch force test using the foot pedal. And in aging, you find that the receptor is not activated as it is in the young. We scored that by looking at the activated receptor by immunofluorescence because it isn't expressed until damage. But the aged have far fewer of these EP4 receptors expressed. They're lower intensity, and altogether the signaling pathway is somewhat disrupted. However, when we isolate the cells and put them in culture and expose them to PGE2, we find that the controls, you see a lot of cell death. The aged muscle stem cells are just not very good at dividing, and many of them die. And compare that with the prostaglandin E2 treated cells by time-lapse microscopy. You're watching in real time these cells dividing. Well, it's a little bit speeded up, but they are real cells dividing. and what you see is that many, many more cells are being generated. And when we quantified that, you can see generation 1, generation 2 of cells accumulating. And what we also noted that we never saw in the young, so many of the aged normally are dying with PGE2. It's not only stimulating them to divide more, but protecting them from cell death. And when we expose them just transiently for 24 hours to the drug and then measure cell numbers later, we see a vast expansion, not quite as robust as in the young, but pretty good. So it says the cells are still able to respond. And interestingly, if we now co-inject PGE2 with isolated aged muscle stem cells, and we look at their engraftment by bioluminescence imaging, we see they're able to engraft. So if taken out of their niche and exposed to this factor, they appear to be rejuvenated in their function. And they are stem cells because they respond to a second injury by expanding. And in another way of looking at the stem cells, we now see that there's an increase in the muscle stem cell numbers, which we ascertained after an injury and doing immunofluorescence for PAX7 positive cells. And this shows the increase in response to PGE2 endogenously. Not only that, but you see an increase in myofiber sizes, and that's a measure of the stem cell function because they're fusing in and making the muscle fibers bigger. So the cross-sectional area is increasing. Furthermore, we took geriatric mice, 24-month-old mice, and we treated them with PGE2, and we saw an increase in their strength just a few weeks later. So this is quite remarkable. A single exposure with a long-term effect on strength in geriatric mice. And then to see whether the receptor was important to the signaling, we eliminated it genetically and aged the mice to about 14 months. That's late middle age, not quite really old, just to see what would happen to their strength. And lo and behold, these stem cells and this signaling pathway are very important even in the absence of any injury. There was no injury. We just let these animals grow and develop. And really it was not quite known that the stem cells are that important to development and maintaining your strength with aging. But I think this was the pièce de résistance. Here we took a mouse, or several mice, and we had them, we exposed them to injury that was a natural injury. Instead of Notoxin, which is the standard in the field, which is a snake venom toxin, which does a lot of damage and nice to measure the effects, we wanted to look at something that you might be exposed to, like downhill running, which is an eccentric exercise that causes muscle damage. So we had the mice run 10 minutes a day downhill on a treadmill, and we gave them PGE2 injections on 5 of these days, and then we had them run for another week. And after 4 weeks, we looked to see what their strength was. And this is what we found. The twitch force increased robustly, and the tetanic force as well. So these are strong measures of improvement in weakness in aged mice. So to summarize, what I told you is that we found an endogenous stem cell activator. It's also a potent, can be used with cells as a cell therapeutic. However, it overcomes a lot of the hurdles to doing a cell therapeutic and is faster, but it could work for both, stimulating endogenous cells and helping with a cell therapy and making it potent for muscle repair and regeneration. And interestingly, PGE2 is required and sufficient by these stem cells. In the absence of the receptor, or if you block the synthesis of PGE2, engraftment fails and mice get weaker. And I think it's fascinating that PGE2 has this heritable effect on muscle stem cell function. We believe it's an epigenetic memory, and we're actively studying how this memory is established in the stem cells and whether it's established similarly in young and aged. PGE2 augments aged muscle regeneration. And so what we're doing here is we are augmenting a natural response. This is the natural healing response, and we're just giving an extra dose of this immune modulator, which could have therapeutic potential. So how can we translate our findings to people? And that's what led me to found this company, Myoforte. I've made a lot of mice strong in my lab with the help of people like Alessandra. And although that's fun, it's the most gratifying thing would be able to translate this to people and to actually help people in the course of aging to use their muscles longer and be stronger and recover, for instance, after immobilization, after a hip replacement or knee replacement. Your muscles atrophy so quickly, especially when you're older. And it would be a dream come true to help with that. So our vision is to rejuvenate the muscle function. There's a large unmet need. Currently, there are no treatments for muscle atrophy in the clinic. And this would counteract aged muscle stem cell dysfunction. So many drugs that are being studied target the muscle fiber to try and make it larger. This is a different approach where we're targeting the stem cells that are fueling the muscle throughout your life. And we're capitalizing on the body's own healing mechanism. We're leveraging the stem cells that the body uses to respond to injury and enhancing the regenerative process. Importantly, for making this successful, we have strong IP of a drug combination and formulation. And I think it has enormous implications for aging. Just to think of a few, carpal tunnel syndrome is extremely debilitating. If you have this muscle wasting in your thumb, you can't do your computer, you can't, if it's really severe, you can't pull up your zipper, you can't hold a fork. So to be able to target this one little muscle and make it stronger could have a huge impact. Rotator cuff tears primarily affect one little muscle, and in talking with clinicians, it looks like this might be one we could target. Incontinence is another problem of aging that is very debilitating to both men and women, and fecal incontinence as well. To make those sphincter muscles stronger, these small muscles would have a huge impact on life. And you can think of eye muscles too. Now what people do is to relax the muscle. If you have a lazy eye, then you make the other one lazy to match it. We would like to strengthen it. We view this as the antithesis to Botox. Botox relaxes everything, we're strengthening it. So, and it might be just as widely used, who knows? That's my dream. So I want to thank the people in my lab who've contributed to this work. I've been so lucky to have a really talented group from all over the world and from different disciplines as well. Without them, this wouldn't be possible. And I want to thank you for your attention, and I'm happy to take questions. Yeah, Katrina.
Helen, that was a fascinating talk as always. I'm feeling inspired. I'm going to not take any Advil and go work out. But anyway, I was thinking when you were talking about PGE2 that Lenzon had a very nice paper in Cell in 2009 suggesting that PGE2 is in part responsive to Wnt beta-catenin in hematopoietic stem cells. Have you looked at the hematopoietic stem cells in these mice where you're looking at PGE2 manipulation during aging?
No, we have not. You're right, that's very nice work that he did to look at cord blood and whether you could use a single cord instead of mixing two. And I don't know where that stands. It seems to have gone quiet. We have not looked at the HSCs. Maybe other stem cells are stimulated by this pathway.
Yeah, very nice.
It certainly seems
exciting.
possible.
Hey, Helen, great talk. So the other muscle problem with inflammation is dystrophy. And so have you thought about, I'm sure you have, what would you think about the output of putting this into a dystrophic mouse model in terms of regeneration or degeneration?
So, good question, Andrew. So Duchenne muscular dystrophy, which is the primary, most debilitating progressive muscle weakness that's inherited, affects children. And one thing we show is that this is aging related, that in order to increase strength, it's primarily useful in the aged. And I don't think I'd want to add inflammation, insult to injury, in the case of muscular dystrophy. So we're not viewing that as our target at all. Other things may work there. A cell therapeutic could. If you treated the cells ex vivo and then you transplanted them, that might be a very viable way to build muscles as a cell therapy, which I think is another avenue worthy of exploring too. What I was talking about is primarily stimulating the endogenous stem cells, and I see that more for muscle atrophy and wasting with aging, or with immobilization and disuse. Yeah.
Amazing talk as usual, very inspiring. I thought very interesting the data with the receptor. So I'm just wondering, when you have the mutation, when you deleted the EP4, what happens to the satellite cells? The muscle is weaker. Do we have less satellite cells, or they're not responding to the injury?
Yeah, the satellite cells. Thank you, Rita. So the satellite cells are still there. They're just not responding, and so they don't proliferate. They just sit there. It's amazing. And you put them in culture and they won't divide. They absolutely need that pathway.
Does the environment, the niche environment in the aged mouse change when you give your drug? So you showed how there was inflammation, and you showed the fibroblast, and you showed that is contributing. I'm just curious, what happens to that environment? And is there an opportunity to even improve it with maybe, I don't know, an anti-inflammatory that's not an NSAID? I mean, or does the PGE2 and the receptor really drive that?
Yeah. So I think that's a really interesting idea, and we're studying this dysregulation, what's gone wrong with the signaling. It's much more complicated than what I said today, because it's still in, we're still experimenting. But we really want to understand how that niche is dysregulated and how you can overcome it. It's not so straightforward, and it's a fascinating problem.
Great talk. So in the cases where PGE2 activity is inhibited by an NSAID or you have EP4 ablation, what happens if you come in and you directly upregulate their downstream target NER1? Does that rescue stem cell proliferation and engraftment? And if so, could you imagine a scenario where you introduce a modified RNA or a vector with a tissue-specific promoter to directly affect the activity of muscle stem cell proliferation?
That's a super question, and we've done that. And if you overexpress NER1, you can rescue the phenotype. So it is another approach that could be used.
Just to get onto the NER1 thing, as you know, NER1 is critical
Yes.
for
Neural cells.
well, for dopaminergic neuronal differentiation, as well as renal, but particularly, have you pursued the other downstream effects of NER1?
Yes, we're looking at the NER1 targets both in young and in aged, and interestingly, they're quite different. And I think therein lies a very interesting story that also relates to the dysregulated niche due to chronic inflammation. So it's a complex story. It's not just cell cycle genes. It's much more than that.
Here. The bottom. Here. The back. Oh. Helen. Is this an effect that extends to other type of adult stem cells, or is it just restricted to muscle stem cells? And if so, why?
So it remains to be explored. And it may well be true for other stem cells. I don't think it's been adequately tested. The hematopoietic stem cells have been looked at. Their response is not as robust as this. And I've never seen anything as robust as this. So I don't know if it'll work in any other cell type. It may be that there's something very specific about muscle
Could be
that's
the
responding
environment.
in this signaling. Right?
Could this be the environment, given the specificity?
Well, when we isolate the cells, we also see that they're robustly, and if you ablate it in vitro, they will not grow. They will not respond to even serum factors, which is quite surprising. They really require this signaling pathway. And then in vivo, I think I showed you with cell therapy and directly targeting the endogenous cells.
Any suggestion of efficacy in cardiomyocytes?
In pardon?
Cardiomyocytes, cardiac muscle?
Oh, cardiac. So cardiac tissue, unlike skeletal muscle, does not have a defined bona fide stem cell. It's hotly debated, but there really is not strong evidence that such a cell exists in the heart, which is a damn shame if you think about it, because we need them in our hearts. We need something to regenerate our hearts and prevent heart failure. But we don't have it, and so PGE2 wouldn't have a target to work on there. But many other tissues do, and it remains to be seen whether this could have similar effects or not.
Hey, thanks for the talk. It looks like in some of the figures, some of the response maybe was associated also with the Notexin injury. So how much do you think the injury environment is sort of requisite for the augmented response? And would you then anticipate sort of different results in like a lack of use or atrophy, trying to regrow that muscle versus trying to regrow an injured muscle?
So injury is key. The stem cells don't express the receptor when they're in a quiescent state. They only express it and can sense that PGE2 is there when there's injury. So you're right on the mark there.
Well, let's thank Helen again for these fascinating talks, and there will be cocktail outside and a reception outside for everyone. So, thank you.
Thank you.